So this is a module 4, that is the carbohydrates. Module 3, sorry, not 4 and 3, carbohydrates. So in this module we will be discussing about the importance of carbohydrates, classification of carbohydrates, how to draw Fisher projection, Haworth structures, and the chair conformation of the sugars. Then we will be discussing about the in details of the proteoglycans and the glycosaminoglycans, as well as the lectins. And many of you have already studied this course. I will try to summarize briefly. And the word carbohydrate is derived from a French word hydrate decarbonate. So you can see here, this is the hydrate decarbonate. That means they are the hydrates of carbon. Hydrate means water, carbon means carbon. So the carbohydrates are the hydrates of carbon. So that is the actual meaning of the French word hydrate decarbonate, means they are the hydrates of carbon. So they are composed of carbon, hydrogen, and oxygen. They are composed of carbon and hydrogen and oxygen. And some sugars, some carbohydrates have nitrogen, phosphorus, and also sulfur. Nitrogen, phosphorus, and sulfur. And these carbohydrates are also known as saccharides because of their sweet taste. And they have the general formula of CnH2O n times. CnH2O n times. So you can CnH2O n times or CnH2O n times. That means, as we said here, that they are the hydrates of carbon. So for example, glucose C6H12O6. So that means this. So glucose follows this empirical formula of CnH2O n times. But certain sugars, mainly deoxy sugars and amino sugars, doesn't follow this empirical formula of CnH2O n times. For example, you can see here deoxyribose C5H10O4. It has the empirical formula of C5H10O4, whereas the ramenose has C6H12O5. This is the deoxy sugar, this is the amino sugar. You can see here, this is the ribose, this is the deoxyribose. Deoxyribose doesn't follow the empirical formula of the CnH2O n times. In the same way, amino sugars like glucosamine, mannose-amine, this is the glucosamine. This is structure of the glucose, and it is the glucosamine, and this is the mannose-amine, and this is the galactosamine, and this is the N-acetyl glucosamine. All these are amino sugars. These amino sugars, especially glucosamine, mannose-amine, galactosamine, if you see, they have the molecular formula of C6H13NO5. So these amino sugars doesn't follow the empirical formula of CnH2O n times or CnH2n and ON. So that means deoxy sugars and amino sugars doesn't follow this empirical formula. And these carbohydrates, we know that they are the abundant molecules in nature, and they are present in humans, animal tissue, plants, in microorganisms, and also the tissue fluids, and also the excretions and secretions of animals. And coming to its medical importance, we know that the carbohydrates are the major source of energy. They are the major source of energy, especially glucose. It is the mainly used as the for deriving energy in the human body. Especially the human brain uses only glucose as the major metabolic fuel because of the presence of the blood-brain barrier. So human brain mainly depends on the glucose. And some carbohydrates are also serve as a reserve food materials. So for example, glycogen, as we have already said, that is a reserve food material in animals, whereas the starch is a reserve food material in the plants. And These carbohydrates are also components in various animals and plant structures. We know that the in animals, especially the skin, connective tissue, tendon, cartilage, bone, all these are composed of the carbohydrate components. This we will explain later when we discuss about the mucopolysaccharide. And in plants, the cell wall has the cellulose, so therefore both the animal and the plant structures are also built with the carbohydrates. And in addition to this, and they are the components in the cell membranes. We have already said when we explained about the biological membrane, and they are the lipid bilayers, and they have the in addition to the lipids, they have the glycan chains on the surface. So therefore these carbohydrates are also components in the cell membrane and also the nervous tissue, and they are the components of the nucleic acids and blood group substances. We know that nucleic acid is a polymer of, they are the polymer of nucleotides, and each nucleotide is composed of the nitrogen base and the either deoxyribose or the ribose as well as the phosphate. So therefore the carbohydrates are also present components in the nucleic acids, and these carbohydrates are also involved in the formation of blood group substances. Blood group substances, what we know, the A blood group or the O blood group or the B blood group, A antigen, O antigen and B antigen, all these blood group substances are formed because of the minute change in the glycan chains present on the surface of the red blood cell. Let us see here, assume that this is the red blood cell membrane. This is the red blood cell membrane, and this is the red blood cell membrane. And this membrane has, if this membrane has glucose, galactose, N-acetyl glucosamine, galactose, N-acetyl N-acetyl galactosamine, and fucose. When the red blood cell has this type of glycan chain on the surface, then it is known as the A antigen. And if it has same structure except N-acetyl galactosamine, then it is a O antigen. And if it has the same structure extra one galactose, then it is a B antigen. That means you can see here, we know that O antigen or O blood group is a universal donor, in the sense it has glucose, galactose, N-acetyl glucosamine, and galactose and fucose. This type of glycan chain is present on the surface of the red blood cell membrane, then it is known as the O antigen. If it has one extra N-acetyl galactosamine, then it is a antigen, and if it has one extra galactose, then it is a B antigen. So that means all these, you can see here, all these blood group substances are differing from one another only because of the minute variation in the sugar residues present on the surface of the red blood cell membrane. So that means these carbohydrates are these involved in the formation of the blood group substances. And in addition to this, we have already said that the sugar chain, the sugars that are carbohydrate residues, what you are seeing here, these are the residues, the sugar residues that are present on the surface of the cell membrane. Cell membrane are involved in various interactions, as we have already said in last class, that cell interaction, cell interaction, cell recognition, cell adhesion, as well as the host pathogen interaction, as well as the host immune system, host immune response, as well as the metastasis and intracellular trafficking and localization. All these are mediated by So, in addition to all these, the glycan chains that are present on the cell surface are involved in various interactions. And students, am I audible? Hello?
Yes, sir. Yes, sir. Now you are audible, sir.
Students, am I audible?
Yes, sir.
So,
So, so that means the glycans that are present on the cell surface are involved in various interactions. Then some of the carbohydrate derivatives are also drugs. For example, you can see here. This is the ovabine. It is the it is the it is a it has the carbohydrate component. You can see here. It is a modified cholesterol, and this is the you can see a sugar component. So some of the carbohydrate derivatives of the carbohydrates are drugs, for example ovapine and the streptomycin, and also some amino sugars are also the components in antibiotics like erythromycin and carbamycin and so on. And so that means the water soluble vitamin is also the water soluble vitamin, that is ascorbic acid, is also the carbohydrate derivative. In addition to this, the certain Antarctic fish survives in the icy environment because of the presence of the antifreeze glycoproteins in their blood. So that means carbohydrates not only the source of energy and also the reserve food material, they have uh, various functions in the cell, are mainly what we have listed here. And coming to the chemical nature, these carbohydrates are polyhydroxy alcohols that we know, because when you look at the structure of the any sugar, we can understand that they are the polyhydroxy alcohol. They have the multiple hydroxyl group either with the aldehyde or the keto functional groups. That means they are polyhydroxy alcohols either with aldehyde or keto functional group in their structure. And these carbohydrates are generally represented with the CnH2O times. This is the formula. We have already said that usually the carbon to water ratio, as we already said that all the carbohydrates are the derivatives of carbon. So that means the carbon to water ratio is always 1. In most of the cases, it is 1. And these carbohydrates are classified into 3 categories. One is monosaccharides. Based on the number of carbon atoms, they are classified into 3 categories. One is monosaccharides. Second one is the oligosaccharides. Third one is the polysaccharides. So monosaccharides have only one sugar residue, and whereas the oligosaccharides are composed of 2 to 10 monosaccharide residues, whereas the polysaccharides are composed of more than 10 monosaccharide residues. And in all these carbohydrates, the saccharose group is present. That's why the name saccharide is mentioned. And these monosaccharides are further classified into different ways based on the number of carbon atoms and also the based on the functional group present. And we know that monosaccharides are the simple sugars. They cannot be hydrolyzed further into smaller ones. And based on the number of carbon atoms, they are classified into trioses, tetroses, pentoses, hexoses, heptoses, and nonoses. If the carbohydrate has only 3 carbon atoms, it is a triose. If the 4 tetros, 5 pentos, 6 hexos, 7 carbons, heptoses, and 9 nonoses, and so on. And based on the functional group, they are also classified into aldoses as well as the ketoses. If the sugar has aldehyde functional group, you can see here, then they are known as the aldoses. If the sugar has the keto functional group, then they are known as the ketoses. If the sugar has 3 carbons and aldehyde group, then it is known as the aldotriose. If the sugar has 3 carbons and keto functional group, then it is known as the keto triose. And you can see here all the see here we have several trioses, tetroses, pentoses, and hexoses. You can see here all the simplest triose is the glyceraldehyde. You can see here this is the glyceraldehyde, and we know that it has the one chiral carbon and one hydroxyl group on the right side. If the hydroxyl group is on the right side, then it is known as the D-glyceraldehyde. If the hydroxyl group on the left side, then it is known as the L-glyceraldehyde. So from all the tetrasaccharides or the pentasaccharides or the hexoses, all these are derived from the D-glyceraldehyde. So D-glyceraldehyde is the source for it. So in case of tetroses and the 4 carbon sugar containing sugars, 5 carbon containing sugars, 6 carbon containing sugars, you can see here that the D-glyceraldehyde configuration is retained throughout the series. And from the we will explain later this series little bit later. And so we have the tetroses, 4 carbon containing sugars, erythrose and threose, and 5 carbon containing sugars, ribose, arabinose, xylose, and lixose. 6 carbon containing aldoses, allose, maltose, glucose, mannose, glucose, idose, galactose, and tellose. So all these are aldoses, aldehyde containing sugars, aldehyde group containing sugars. Then coming to the keto group containing sugars, these are also known as ketose family, and the simplest ketose is the dihydroxyacetone. You can see here dihydroxyacetone. And from the dihydroxyacetone, again, when we introduce one more carbon, it becomes erythrulose. From erythrulose, when we introduce one more carbon, it becomes ribulose and xylulose. And ribulose, we get again, after introducing one more carbon, psychose and the fructose. From the xylulose, sorbose and tegatose. We will see how to remember these structures later. And before explaining about the in details of the aldose sugars, the aldose family as well as the ketose family, let me give you brief introduction about the properties of monosaccharides. We know that all these monosaccharides, they have the one or more asymmetric center except dihydroxyacetone. So in the sense, the asymmetric center in the sense carbon that is bonded with the 4 different atoms or the 4 different groups are said to be asymmetric center. And these carbohydrates except dihydroxyacetone, all monosaccharides have the one or more asymmetric centers. And sugars also exhibit various forms of isomerism, that is stereoisomerism as well as the optical isomerism. We know the stereoisomerism in the sense they have the same molecular formula and the same structure, but the different spatial configuration. And you can see here, all these are all these isomers are the stereoisomers. They have the, for example, they have the same molecular formula and the different for example, glucose and galactose, they have the same molecular formula, but the different spatial configuration or the spatial arrangement of the atoms. So that means that these are so all monosaccharides exhibit the stereoisomerism as well as the optical isomerism, and all these monosaccharides, because they have the asymmetric carbon, and all these monosaccharides can have the various isomers, and the number of isomers depends on the number of asymmetric carbon, and therefore the number of optical isomers of a given sugar depends on the number of asymmetric carbons present. For example, if the sugar has the 4 asymmetric carbon atoms, and the number of possible optical isomers is 2 power n, where n is equal to number of asymmetric carbon, that is 2 power 4, then becomes the 16 isomers. So that means, according to the rule of n, or the Liebel-Van 't Hoff's rule, the number of optical isomers possible for a compound is equal to 2 power n, where n is equal to number of chiral centers. In this case, glucose, it has the 4 chiral centers, 1, 2, 3, 4 chiral centers, and therefore it so since all monosaccharides have asymmetric carbon atoms, so they rotate the plane polarizer light, and if the sugar rotates the plane polarizer light towards the right, then it is known as the dextro, and it is also designated with the small d or plus sign. And if it So that means here the capital D indicates that is the capital D doesn't say anything about whether a sugar is a dextro or the levo. Capital D indicates the configuration of the sugar at the bottom stereocenter. You can also see here whether if the hydroxyl group is on the right side, then it is known as the D, and if the hydroxyl group is on the left, then it is known as the L. Capital D and capital L indicates the configuration of the sugars, and whereas the small d, small l indicates the optical activity. Small d indicates the dextrorotatory, and the small l indicates the levorotatory substance. And we know, and coming to the enantiomers and the diastereomers, we know that the stereoisomers, in the sense they have the same molecular formula and the same structure, but the different spatial arrangement of atoms. But enantiomers are the stereoisomers that are mirror images of each other. You can see here D-glucose and L-glucose, they are mirror images of each other. And in the same way, you can see here the compound 1 and the compound 2, these two are mirror images of each other. So stereoisomers which are mirror images of each other are enantiomers. Stereoisomers which are not mirror images of each other, they are the diastereomers. You can see here 1 and 3 are the diastereomers, and 2 and 4 are the diastereomers. So in the sense, they are not mirror images of each other. Then epimers, if the two sugars differ in their configuration around only one specific carbon atom, they are said to be epimers. For example, see here glucose and galactose. And glucose and galactose are epimers in the sense the glucose, this is the structure of the glucose and this is the structure of the galactose. So they differ in the configuration 1, 2, 3, 4 at the fourth carbon. On the fourth carbon, glucose has hydroxyl group on the right side, whereas the galactose has the hydroxyl group on the left side. So that's why they are known as the C4 epimers. So in the same way, glucose and mannose are C2 epimers. You can see here, this is the glucose and this is the mannose. They differ from one another at the second, so that they differ from one another only at the carbon 2. They are known as the C2 epimers. And you can clearly see here, this is the glucose and galactose, they are C2 epimers. Glucose and mannose, they are glucose and galactose C4 epimers. Glucose and mannose, they are the C2 epimers. They differ from one another only at the carbon 2. And now coming to the before going to explain about the cyclic structure of the monosaccharides, I just wanted to make it clear that D and L sugars, capital D and capital L sugars, are the DL configuration of sugars. If the sugar has same configuration as of D-glyceraldehyde, so this is the D-glyceraldehyde. Look at here, this is the CHO, this is CH2OH, and this is OH, and this is H. This is the D-glyceraldehyde. If the sugar has D-glyceraldehyde configuration on penultimate carbon, then they are known as the D sugars on the penultimate carbon, in the sense last but one carbon. So for example, see here D-glucose. This is the carbon 1, 2, 3, 4, 5, 6. This is CH2O, OH. This is CHO, and second carbon, third carbon, fourth carbon, fifth carbon, OH, OH, H, OH. Again H, H, OH, H. So you see here, if the sugar has D-glyceraldehyde configuration on penultimate carbon, that is nothing but the last but one carbon. This is the last carbon, this is the first carbon, 2, 3, 4, 5, 6. So on penultimate carbon or the last but one carbon, if the sugar has the D-glyceraldehyde configuration on the last but one carbon, then it is known as the D sugars. If the sugar has D-glyceraldehyde, L-glyceraldehyde configuration on the penultimate carbon, they are known as the L sugars. For example, we know that L-glyceraldehyde has the hydroxyl group on the left, and this is the CH2OH, this is L-glyceraldehyde. If the sugar has L-glyceraldehyde configuration on the penultimate carbon, they are known as the L sugars. For example, if the hydroxyl group is on the right side, then it is known as the L-glyceraldehyde. So D sugars in nature, D sugars are the abundant. So that's why we have mentioned here all the only D sugars. In the sense, only D sugars are naturally abundant, not the L sugars. So that's why all these are D, all these sugars, that is 4 carbon containing sugars, or the 5 carbon containing sugars, 6 carbon containing sugars. In all these, you can notice that the penultimate carbon has the hydroxyl group on the right side. Penultimate carbon has a hydroxyl group pointing on the right side. So now we need to understand how to remember these aldose sugars as well as the ketose sugars. So the simplest way to remember the simplest way to remember these aldose and ketose sugars is that, so we need to remember first only the D-glyceraldehyde structure. So as I already said that all the D sugars are derived from D-glyceraldehyde, that is CHO, single bond COH, single bond H, single bond CH2OH. This is the D-glyceraldehyde. So now, in order to write the structure of the all D sugars, you need to remember the simplest shortcut, that is you can, I am just writing here GET, get Raxel, AA, GM, GI, GT. So this is the shortcut you need to remember in order to, instead of memorizing all these structures. So when I say here, if you know the glyceraldehyde structure, D-glyceraldehyde structure, we can write any sugar from glyceraldehyde. When we increase the 1 carbon, we get erythrose and triose. From erythrose, again, after introducing 1 more carbon, we get ribose and arabinose. From triose, we get the xylose and lixose. From ribose again, we get the, when we introduce 1 more carbon, we get allose and maltose, arabinose, glucose and mannose. From xylose, glucose and idose. From lixose, galactose and tellose. I will just show you one. So I am just first, what you need to do is first you need to write the glyceraldehyde structure. I am just writing the glyceraldehyde structure. So this Fischer projection, CHO, this is the OH, and this is the H, and this is the CH2OH. This is the glyceraldehyde, that is the 3 carbon containing sugar. So you can see here, when we introduce 1 more carbon, to the glyceraldehyde, again write the glyceraldehyde structure here once again, CH2OH, again here OH, H, again here CHO, CH2OH, again OH and H. So this is the D-glyceraldehyde. So when we introduce 1 carbon here next to the aldehyde functional group, see here, next to the aldehyde functional group, 1 carbon, and place hydroxyl group once on the left and once on the right. When you place hydroxyl group once on the right, once on the left, you get erythrose and triose. You can see here, you get D-erythrose and D-triose. So when you place hydroxyl once on the right, then it becomes H. When you place hydroxyl once on the left, then the right side becomes H. So then this is D-erythrose, and this is D-triose, D-erythrose and D-triose. Now From D-erythrose and D3-ose, again from D-erythrose, again when you introduce one more carbon next to the aldehyde functional group and place hydroxyl once on the right and once on the left, then you get ribose and arabinose. From 3-ose, again when you introduce one more carbon next to the aldehyde functional group and when you position hydroxyl once on the right and once on the left, then you get again xylose, lixose. And from ribose, again when you introduce one more carbon, again place hydroxyl once on the right, once on the left, then we get allose, maltose. Again in the same way, from the arabinose, glucose and mannose and glucose and idose, galactose and tellose and so on. So what you need to remember here is that you need to remember the D-glyceraldehyde structure. So D-glyceraldehyde structure. So when we introduce, this is the new carbon. See here, this is the new carbon. So when we introduce the new carbon next to the aldehyde functional group, and when you place once hydroxyl group on the right, once on the left, then it becomes a D-erythrose and D3-ose. Again, from this, when you draw the same structure of D-erythrose, again introduce one more carbon next to the aldehyde functional group, again introduce hydroxyl group once on the right, once on the left, then it becomes the D-ribose and D-arabinose. So from the D-ribose, when you introduce again one more carbon and place hydroxyl group once on the right, it becomes a D-allose. When you place hydroxyl once on the left, then it becomes a D-aldose. So remaining structure is the same. You can see here. What we are doing here is when you want to write the aldose sugars, you need to remember only the D-glyceraldehyde structure. And from the D-glyceraldehyde, when you introduce one more carbon, and when you get the D-erythrose and the D3-ose, from the D-erythrose, again when you introduce one more carbon and place hydroxyl group once on the right, once on the left, then you get the D-ribose and D-arabinose and so on. For example, let us let me tell you. Let me tell you one example. I want to write the structure of the glucose. So as we know that we have the shortcut method, that is the get Raxel AA GM GI GT. This you need to remember. So from glyceraldehyde, erythrose to ribose and arabinose, ribose to allose and maltose, arabinose to glucose and mannose. So I want to write the structure of the glucose. You can see here, write the structure of the glucose. I need not remember the glucose structure. What I need to remember here is that if I remember the structure of the glyceraldehyde, D-glyceraldehyde, that is the D-glyceraldehyde, CHO, CH2OH, and OH and H. This is the D-glyceraldehyde 3-carbon containing sugar. But I want to write the glucose structure. So what you need to do is again write the glyceraldehyde structure once again, CHO, CH2OH, because I already said that all D sugars have the glyceraldehyde configuration on the penultimate carbon. So this is the glyceraldehyde once I have again written here. So now when from 3-carbon, when the glyceraldehyde, when you introduce one more carbon next to the aldehyde functional group, and when you place it hydroxyl group on the right side, then you get the erythrose. Again, that is, it is a tetrose. When you introduce one more carbon, again place hydroxyl group on the right side, again we get ribose. This is the structure of the ribose. Again, see here. So if you place hydroxyl group on the left instead of right, then we get arabinose. And again, from the arabinose, again when you introduce one more carbon and place hydroxyl group once on the right, then we get glucose. So this is the structure of the glucose. OH, OH, H, OH, CHO. In the same way, H, H, OH, again H. This is the structure of the D-glucose. When we say D-glucose, when we say D sugar, all the D sugars, you can notice here that the penultimate carbon, that is the last but one carbon, has the D-glyceraldehyde configuration. In the sense, hydroxyl group is pointing right. In all these sugars, hydroxyl group is pointing on the right. That's why they are known as the D sugars. So this is the simplest way to write the any sugar, using the without memorizing, by simply remembering the structure of the D-glyceraldehyde. If you know the D-glyceraldehyde structure, if you remember this shortcut, that is the get Raxel AA GM GI GT, and you can see that how the tree is getting elongated from the glyceraldehyde to erythrose and threose, from erythrose to ribose and arabinose, from ribose to allose and maltose, from arabinose to glucose and mannose. In the same way, from threose to xylose and lixose, from xylose to glucose and idose, from lixose to galactose and tellose. So this is this you need to remember, then only you will be able to write the structure of the any sugar. You need not memorize the structure of all these sugars if you need to remember only the structure of the D-glyceraldehyde. In the same way, you can also remember the structure of the. You can also write the structure of the keto sugars. As I already said, that keto sugars have the keto group. The simplest ketose is the dihydroxyacetone. So this is the CH2OH, and another side CH2OH. So this is the dihydroxyacetone. So this is the 3 carbon containing the ketose, dihydroxyacetone carbon, acetone. And you can see here, when we introduce one more carbon, see here, one more carbon. So from the dihydroxyacetone, when we introduce one more carbon next to the keto functional group, and when we place hydroxyl group on the right side, then it becomes a D-erythrose. And again, after that, when you introduce one more carbon here, and place hydroxyl group once on the right, once on the left, it becomes D-ribulose and D-xylulose. From D-ribulose, again introduce one more carbon next to the keto functional group. Again here it becomes a D-psychose and it becomes a D-fructose. In the same way, from xylulose, when you introduce one more carbon next to the keto functional group and they place hydroxyl once on the right, once on the left, then you get the D-sorbose and D-tegatose. So that means from the dihydroxyacetone, we get first D-erythrulose, then the D-ribulose and the D-xylulose. From D-ribulose, we get the D-psychose and D-fructose. From the D-xylulose, we get the D-sorbose and the D-tegatose. So in the sense, in order to write the structure of the D sugars, so in order to write the structure of the aldoses, we need to remember the structure of the glyceraldehyde. In order to write the structure of the any ketoses, we need to remember the dihydroxy ketone. And of course, for this also you can remember this shortcut, that is the DE-RX PF-ST. DE-RX PF-ST. That means from the dihydroxyacetone we get erythrulose, from erythrulose ribulose, xylulose, from ribulose psychose and fructose, from xylulose sorbose and tegatose. So this also you remember DE-RX PF-ST. So if you can remember this shortcut, you can able to write the any the ketose structure if you know the dihydroxyacetone structure. So you need not memorize anything. For only thing is for writing the aldoses, you should know the D-glyceraldehyde structure. For writing the ketoses, you should know the dihydroxyacetone structure, such that you will be able to write the whether the aldose series or the aldose family or the ketose family sugars without memorizing. So this is the way we need to write the structure of the monosaccharides. So I think in afternoon class we will discuss about the cyclization of sugars. I think it is already 1 o'clock.